Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 137th seminar, and I call it on ONR Global Day. I am in the United Kingdom to give 3 invited talks at Imperial College London, UCL. Actually, today I just finished my talk, and University of Cambridge next week, followed by 2 talks at 2 conferences in UK and also Germany. It's great to meet with my colleagues and new people here in Europe, including tomorrow I'm meeting also editors of Nature Biotech and also Trend in Biotechnology, of course, Self-Press. And then I really enjoy the meeting a lot of people here and such a nice, continent to talk about, science and engineering. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Martina Siwek. She is a regional chief scientist at the Office of Naval Research Global, in short, ONRG. I met her in 2022 during the Serendipity Collective competition to brainstorm and compete with other innovators in Berlin, Germany. At the time, she kindly helped my team prepare for the competition, idea development, and presentation. By accident, she and I took the same train in the same car to go to Prague, Czech Republic, where her office is located. I enjoyed my short vacation there, and she gave nice tour information and tips where to go, where to eat, etc. So Martina, thank you so much for your, such a nice tip, 2 years ago, and that is a memorable trip for me. And also thank you so much for your global leadership and the contribution to the scientific community, and the virtual podium is all yours now. And thank you again.
Thank you, Taesuk, for that kind introduction. It was definitely my pleasure to work with you in Berlin, and I'm so glad you enjoyed your time in Prague. So as you mentioned, I do work for the Office of Naval Research Global, ONR Global, and I'm located in our Prague office. And today I'm going to take the opportunity to speak with the audience about interesting Department of Defense and other career opportunities that they may not even know exist, that I certainly didn't know existed until I was contacted by a headhunter that said, "Oh, we have this job." And I was like, "What? What is this job? Oh, that sounds really interesting." And it's my pleasure to be joined by Dr. Lee as well, and I'm looking forward to his discussion later today. So let me just get started really quick. I'm going to share my screen, and I look forward to any questions later. Let's see. Okay. Let me know if you can see.
Yep.
All right, perfect. So I'm going to start with a little bit of information about ONR Global, who we are, what we do, how we operate, and then talk about how you could potentially join either through a collaborative effort with me or one of my colleagues, or even potentially looking at a future career opportunity for young students. So the Office of Naval Research Global is, as it says in its name, a global organization. We have offices in Prague, Czech Republic, where I'm located. We have offices, our headquarters are in London, where Taesuk is right now. We also have an office in Santiago, Chile, in Sao Paulo, Brazil. So those are South American offices. And then we have an office in Tokyo, Japan, in Singapore, and also in Melbourne, Australia. So we have offices in many areas of the world. We just opened an office in India. We're really excited about that. And we are looking at how can we open an office in Africa as well, so that we have much of the globe covered. Right now, we cover Africa through our European offices, but we're really hoping that we'll be able to actually have an office on the continent at some point. So just a little bit about what we do and what might be interesting career opportunities. Myself, I'm the regional chief scientist, but essentially what I do is a role in our international science program. So I scout technology. I get to travel the world to look for very interesting science and research, whether that be at the very fundamental level, so with researchers like Dr. Lee or like Tae-Suk, or with startup companies that already maybe have a little bit of an idea, but they're still very heavily engaged in that basic fundamental research. And so the role I fill is what we call a science director. You can see that on the slide on the left hand, well, for you, that might be your right hand side, or the International Science Program. And you can see all of the offices there as well in the International Science Program. So we really engage in looking for those disruptive, interesting, innovative, blue sky ideas that are really potentially crazy and out there, but we think, oh, wouldn't it be really fascinating if that worked? And we could be at the very start of that type of research. So we look for those researchers that are doing that kind of research. And my particular area of expertise is molecular microbiology and immunology. But I also have a regional focus. And so we both travel and look for technologies in our technical area, but also in our regional areas. And so we kind of go back and forth between that. We also have a group of individuals, which we call the science advisors, who are more focused on the here and now. What are the technologies that are already available, that are already fieldable, and which we could potentially take advantage of today? Then we have a group that is called our International Engagement Office, and they paved the way for us by putting in place a lot of agreements and different mechanisms, memoranda of understanding, so that we can partner with our international organizations and partners. We do have a foreign comparative test group as well that's engaged in actually testing in the field those technologies which are really almost fieldable or already commercially available. So those really high technology readiness level or already on the market type technologies, to see if they wouldn't be a good fit for our organization. And then we have two groups that are experimentation and analysis and tech solutions that take the needs of the sailor or the Marine that we support and try to look for more rapid solutions. So that's more of a demo or an experimentation type event. And then our London Tech Bridge is an interesting and slightly different component to our organization, which really partners very closely with the UK, and they look for needs that are a combination of US and UK. And so they look for technologies that will fill the needs of both nations. And so that's really what ONR Global does. We are technology scouts, and we look and try to deliver those technologies because we're interested in them. We try to get that knowledge out there. We publish our results, or we have our PIs, I should say, publish their results. And so we're really looking to expand that breadth of knowledge and awareness and those capabilities as much as we can. And so that's ONR Global. Before that, I worked actually for a group called the Administration for Strategic Preparedness and Response, or ASPR. This is actually a US Health and Human Services organization. So unlike ONR Global, which is a Department of Defense organization, this is a Health and Human Services. And the group I worked for was the Office of Innovation and Industrial Base Expansion. And they really looked to bring capabilities to the US and to be able to have some of that capability within the US. And so they're trying to expand and build that base for having pharmaceuticals on demand, or for instance, this was heavily used during COVID. That's why I joined at that time. I'm a virologist by training, and respiratory virologist, in fact. And so during COVID, there were just so many technologies that we needed. And so it was an ideal opportunity to really be able to leverage my skills working for the Health and Human Services in this more of like emergency response type situation. What would we need if, say, we didn't have power? Or what would we need, some of the questions with the vaccine, they needed a cold chain. How do you maintain that if you don't have a cold chain? How do you get needles if you don't have access to needles and you need them for the vaccines? Those types of things. And again, this is an organization that has both government employees and contract employees. So you don't necessarily have to be a US government employee to work for this organization. They do have quite a few contract employees that work with this organization as well. The other type of organization that not many people are familiar with are called UARCs, University Affiliated Research Center. So I specifically worked for the Johns Hopkins Applied Physics Laboratory, which is why that one is highlighted on the slide. And it's a Navy UARC. So you can see it's one of the blue ones. It's actually located here in Maryland. However, there's a Navy UARC in Pennsylvania, which is also the University of Pennsylvania Applied Physics Laboratory. There's one at the University of Washington. There are Army UARCs. There are just Department of Defense UARCs, Strategic Command, NSA. And these are organizations within a university that many people who attend these universities don't even know exist. And they actually serve the organization that they support. So for instance, the JHU-APL was a Navy UARC. So they have to maintain certain capabilities and competencies in the staff that they hire to fulfill the specific requirements that the Navy has laid out and said, we need experts in this engineering technique, in this and this and this. And so these UARCs are really geared toward hiring very technical, specifically trained experts in various fields in order to be able to provide the support for the projects that whoever they are aligned with requires. And there are thousands of scientists that work for these types of organizations. This is not a government organization, but it supports the government. Then I also worked for what we call the Global Emerging Infection Surveillance Group within the Defense Health Agency. And this is an interesting group because many people don't really know that the Department of Defense has laboratories, research and surveillance laboratories all over the world. Most people assume that the US Department of Defense only has labs in the US. It's actually not true. They have labs scattered around the world. You can see them with the little stars here on this briefing. And GEIS, the Global Emerging Infection Surveillance Group, is kind of like a mini NIH for these laboratories. And they fund the surveillance activities that these laboratories carry out. So these laboratories work very closely with other local laboratories to conduct the surveillance, the research, the activities. However, the work that they're doing is funded by the US government, by this GEIS group and by several other groups as well. So there is one in Korea, actually, the Public Health Lab in Korea, and there's also the 65th Med Brigade. Those are both located in Korea. And I've been able to visit, actually. They do some very interesting work, and they partner with a lot of Korean universities as well. And so, again, this is another opportunity where you can shape what science is conducted around the world through as I said, kind of like this mini NIH funding for the Department of Defense laboratories. And then the last groups that I'm going to talk about is the pipeline for technology development within the Department of Defense. So I'm going to start with DARPA, the Defense Advanced Research Projects Agency. This agency is really focused on technologies that I'd say are 10, 20 years out. So really the far forward, what are going to be our needs in that many years, and what kind of technology solutions might we have ready in that 10, 20 years to be able to serve the needs of the future. So that's the first step in Department of Defense technology development. The second step is DTRA, the Defense Threat Reduction Agency. They do more the 2 to 5 year out technologies. So a little bit more higher technology readiness levels, and a lot of the products that they develop are focused on chemical, biological, radiological, and nuclear defense. So how do we detect and protect against those types of issues or challenges within our national security? And then the third group and the final group, which is more the acquisitions. Okay, now once you have a ready technology, how do you actually get it out? How do you field it? How do you hire it? How do you make sure that you have enough? So the JPEO, the Joint Program Executive Office for Chemical and Biological Defense, is really engaged in that, okay, we have these great technologies. How do we get them out there? How do we provide them to the people who need them? And so there are a lot of acquisition, fielding, life cycle of technologies. And so those are all somewhat related, and they have a pipeline of technologies that go from, very early research all the way into, okay, now we have it fielded and ready to go. And these are just mostly Department of Defense job opportunities that, like I said, when I was in graduate school, I didn't know existed. And so for those who might be interested in thinking, well, maybe I'm not sure if I want to work in an academic lab, these are some other opportunities where you could still work in a lab potentially, or in a program office, but it may not necessarily be in academia or private industry. It's a totally different avenue that's available. Thank you so much for your time. I really appreciate it.
Excellent talk. I even didn't know there are too many other things exist except for ONR Global and DAPA and DITRA. So thank you so much for informative, explanation and the presentation. That's very useful, will be useful for many audience, for this, seminar. Thank you so much.
Thank you for the opportunity. I really appreciate it.
Thank you. Okay, now the main speaker of today with a longer introduction. Dr. Jeong Wook is a professor at the Pohang University of Science and Technology, in short, more known as Postech. He received his bachelor and PhD degree from Korea Advanced Institute of Science and Technology, also in short, KAIST, in chemical and biomolecular engineering, under the guidance of Professor Sang-Yup Lee, one of the pioneer speakers of this seminar series. He was a postdoctoral research fellow at Boston University, MIT, and Harvard University under the guidance of the Jim Collins, another speaker of this seminar series, and also the synthetic biology father. His research focuses on constructing genetic circuits for cell-based or cell-free gene expression regulation, including applications in metabolic engineering, genetic biocontainment, molecular diagnostics, and molecular biomanufacturing. Importantly, he is a very kind person, and every time when I visit Korean conferences or his university for invited talks, he always treats me very nicely with the best Korean cuisine at a fancy restaurant. So I still remember all the thing I tasted over there. Also, more importantly, his paper with Jim Collins is one of my favorite papers that I used to use for my class in the university I used to be. So Jeong-wook, thank you so much for your time today and contribution. And please take it away, all yours now. And thank you again.
Thank you so much, Tessa, for this really kind introduction. Also for organizing this meeting every time, providing me and all of us with this great, opportunity to engage with this wonderful community. I'm really honored today and also really excited to talk about what I'm working on. Also like to have some discussions with all of you. Okay. And I think it's shared, I guess. And so what I'm going to talk about is design and implementation of gene expression control device. And the first thing I'd like to share is, first of all, I'd like to share what is the boundary that I'm interested in. Also, then I'll move into how to design, construct, and utilize genetic circuits. Then some of the cell-free application of the circuits for developing new molecular diagnostics. And so this is the ecosystem around us. There are many life forms like plants and birds and fish and even not quite visible, these microorganisms. And the boundary that I'm interested in is harnessing this kind of biological behaviors and underlying the molecular reactions in these organisms. And by doing so, developing an efficient tool to achieve this kind of the acquisition. Most of the biotechnology falls in this kind of category, but when we isolate these bioresources from the nature, so these isolated biological resources from nature are not necessarily a perfect fit for our purpose. So what we need to do is we need to modify this a little bit to accelerate or enhance their functionality in a biological, depending on the type of the biological application, we need to modify or we need to adjust it a little bit. And I think the genetic circuit that will be helpful in doing that, because the circuit can operate as we instruct them in cell, how to instruct in cell or biological systems. So in our lab, what we do is we design and construct the circuits, and we apply this to biomedical application, environmental application, or even for the cell-free system, we apply this to molecular diagnostics. And for cell-free system, we also get some help from this cell-free system. We acquire and we also prototype these biological components required for these genetic circuits. That's basically the boundary that we are working on. And then what's genetic circuits? Circuits in short term is an expression control device. And this is the genetic network, and the red dots indicates the protein. The lines indicates the relationship between them. It looks quite complicated, but if we expand this part and enlarge it, then it is looks composed of activation regulation and repression regulations. So genetic circuit is a combination of this. Not just combination. This is a kind of the purposeful combination of this all the regulation. That's a genetic circuit. And typical example would be the microwave kill switches. So by having this kind of circuits and connecting them to the toxin expression or the essential gene repression, then what we do, for example, we regard this ATC, the inducer, common inducer, as a survival signals. If we remove this signal, or maybe if the cell moves into other environment that doesn't have this ATC, then cell will rapidly die. that operation can be programmed by having this kind of the circuits. And this is the only single input cases. If we expand this to multiple inputs like A and B and C inducers, for example, then what this represent is that only in the presence of A and B, not C, and only we have A and B, but not C, cell can happily grow. With all other conditions, including nothing included or everything, every combination included, cell will die. That can be done with this having this kind of the circuits. And then when we apply this to using different signals, then we were able to get the cell survival in the presence of IPTG and galactose, but not cell lobules in these conditions. That's what we can obtain using this kind of circuit. And by having that kind of the circuit controlling gene expression of either essential gene or maybe expression of toxins can only protect the engineered organisms. But there are many other resources we need to protect in the environment. So we thought maybe if there's a way to control from the genetic sequence protection level and genetic material level and plus the engineered organism levels, then we might that system, that kind of biocontainment system would be more useful. So we came up with a biocontainment system based on quadruple codon. So that how that works is that logic is simple. In the presence of the quadruple codon in a protein, and in the presence of this quadruple codon decoding qtRNA, in the presence of this, having 4 different quadruple codons, cell can express this protein. But in the absence of this, then the normal typical triplet codon comes in, and then we gonna have a soon truncated or mistranslated protein. That's the logic. So by controlling the expression of the quadruple codon decoding tRNA, qtRNA, that's what we call here from now on qtRNA. Having this qtRNA, we can control the expression of such protein. So where we can apply this to is to control first genetic traits, like if there's any enzyme converts the one substrate into product, then we insert the quadruple codon there in the middle of the sequence. Then in the presence of this qtRNA, we can have a proper enzymes. But if we don't express this, then we cannot have this protein so that we can protect this kind of the genetic traits. And also we can apply this to control the plasmid replication. And plasmid replication case, there are the initiation factors involved the plasmid replication mechanism. And this is a TRFA gene. And if we insert in the middle of the TRFA gene, in the middle of the gene, we insert the quadruple codon. And by controlling this qtRNA, in the presence of this, we can replicate the plasmid in the cell. Without this expression, the plasmid will not be expressed. And the same thing applies to the essential gene. When we put the quadruple codon in the middle of the essential gene, then in the presence of this qtRNA, cell happily grow, replicates, but without that, cell cannot survive. That's how we can control this. And I think the beauty of this system and the most advantageous part would be it is easy to combine different layers of the protection. For example, by only inserting this quadruple codon to a proper site in the middle of the functional enzyme or in the middle of the plasmid replication initiation factors, or maybe in the essential gene, we can control either cell level control, plasmid level control, and genetic traits level controls. And when we combine these all three together, we were able to tightly control the phenotype as well as the cell growth. And when we have each second levels and only one levels, we can protect, but at the same time, we can have all that together to protect 3 different layers all together. That's pretty much we do for developing new genetic biocontainment systems. And let's go back to the circuit stories. And one of the work that we are doing is designing circuits. And I brought the passcode example again. And when you have a certain kind of the operation that you want make cells to do, then you can probably summarize that into this kind of tables. So A and B and C, that's input, and X, that's output. So in this passcode case, this is the combination of the inputs, and X would be the expression of the toxin. So in the expression, 1 means the expression of the toxin, meaning that cells die, and the 0 indicates there's no expression, makes the cells survive, meaning that only in the presence of A and B, but not C, cell will survive. And once you have the table, then what we are doing to design the circuit is just generate this kind of a circuit. That's the point that we are working on regarding the circuit design. And there are famous, well-known circuit design software called Cello by MIT Chris Boyd group. Our approach is slightly different. We are aiming, first of all, to many different non-model organisms. Another part is because we are targeting that, and we need the expanded pool of the genetic parts and modules. Because we need a lot, we are also trying to develop new approaches for obtaining different genetic parts and modules for non-model organisms. That's what we are doing. And this is the typical the design workflow for designing the circuits. And once the truth table is provided, then we use a software called YOSHIS that's designed for making electric circuits. And we can, by putting this information to the YOSHIS, then we can generate this kind of the logical trees. So once we have this, but up to here, there's nothing biological. What we do is we put biological part instead of this electric part here to find which combination of biological part gives the proper truth table answers. That's what we do. And once we assign which gate goes where, then based on that, we can have the string of DNA informations. That's basically circuit design procedures. And to connect this biological behavior to computer understandable world, what we do is we evaluate each gate, that's a gene expression cassette, each gate, logic, each gate. We evaluate each gate using x-axis as an input signal, y-axis as a fluorescences. We measure this and we acquire this response curve for each gate. And this is a typical the gate, and also represented by Cello's case. And we also recapitulated that the experiment, and were able to get this kind of the parameters based on this empirical Hill equation. And based on that, so these kind of parameter sets becomes now identity of this each different gate. So that having this and having that values, and by combining those mathematical models, then we were able to get which one would be the more appropriate to generate that circuits. That way we can have the DNA sequence information. And by doing so, because we are targeting non-model organisms, we need also different kinds of the transcription factor, alternative transcription factors. So one good alternative is CRISPR-I. And as you can see here, when we use this CRISPR-I as repressors, we were able to achieve nice these parameters as well, meaning that this can be used as a good alternative for transcription factors. And also, we are also building a method to find the good single guide RNA to be used in such a decast system. So we target transcriptome, mRNA of the different organisms. So what we do is our method, this is the method to generate a single guide RNA library, especially for those not well characterized things. Then what we do is we culture the cell and obtain the mRNA of the strain, and we chop down to the good lengths. Then we apply different series of different enzymatic reactions to achieve this proper length and proper sequence of the guide RNA library. Then we can apply this to find the essential genes or auxotropic system, and some phenotype, some gene combination to find the good phenotypes as well. And also this is some of the result with GFP, and when we made the library of the GFP guide RNA, we were able to find which this one works for the single gene, not only single genes, also we can apply this to transcriptome levels as well. Also, not only the genetic modules, gene expression cassette levels, but also we also trying to find the good ways to collect different kind of genetic parts as well. And we are trying to use a cell-free system. And as you know, cell-free system cannot exactly reflect what's happening inside of cell, but it can be used as a rough mirror reflecting what's happening. So it can be used as a good tools to estimate the events in the cell. So the example that we used to generate different kind of genetic parts, in this case promoter, was cyanobacteria. As you know, cyanobacteria, it can grow in the presence of light and CO2, and a little bit of mineral can generate oxygen and the sugars. So it's a good system, right? But it grows slowly, and it is difficult to transform and doing selections. And so if we are trying to build different promoters, we need to, in a traditional way, we need to transform one by one to see the effect. But if we can make a cell-free system somehow, then we can evaluate and we can avoid of these transforming procedures. So what we did was we culture the grow the cell and collect the cell and lyse them, and also optimize the components a little bit. We were able to create the transcription system for cyanobacteria. Then using that, what we did was we put DNA fragments containing promoter-like sequences. Then that way, if the promoter-like sequence works, then it will transcribe whichever the reporting information. So for the reporting information, we use a CRISPR guide RNA for this Cas12a. So what we did was, interesting thing about this Cas12a is that it also is used as a platform to detect the nucleic acid as a molecular diagnostics called a detector. But at the same, this nuclease activity can be utilized differently as well. So how this enzyme works is that in the proper combination of the three, for example, in the presence of the template, if this guide RNA finds the proper template and it will form a complex together with the Cas12 enzyme, then it will cut the proper cutting site. But this entire complex becomes a collateral nuclease activity. It shows collateral nuclease activity. So once we put this kind of probe having one side quencher, the other side fluorophore, then it will cut the DNA and it will fluoresce. That's how it works. So if there's no guide RNA formed, then nothing happens. But by the action of the promoter, it expresses some amount of the guide RNA, then it'll cleave this proof so that it'll fluoresces. So This is the dotted line indicates the RT-PCR, the bar graph indicates our new method, and we found that somehow this correlates with the existing method. So what we did was, using that way, we evaluated the promoter of the cyanobacteria. I mean, that this is actually TRC promoters, which has been used cyanobacteria for quite a lot. So what we did was we tried to characterize these promoters. The logic here is that when we mutate the important region of the promoters, then probably the expression level changes will happen a lot. That's the logic we had. So in this case, 100%, that's the normal activity. As you might expect, -10 region and -35 region regarded as an important area. So when we mutate that area, then we were able to see a large decrease in activity a lot, -35 as well. So we took some of these samples and evaluated that in vivo, and when we correlate the in vitro activity versus in vivo, we can have the good correlations. Then what we did was, as I said at the beginning, we want to have different parts easily, right? So to generate the different parts, so this is the evaluation method, the cell-freeze evaluation method. So what we combined together with was AI technologies. And so first of all, what we did was we made generative models that generates lots of cyanobacteria-like promoters. And the data that we used was a tRNA sequencing result. In this sequencing result, what we can have is that where is the TSS site, so that we can extract the promoter sequences from there. So by using that as a training set, we were able to make a nice VAE model to generate the cyanobacteria promoters. Then what we did was, using the same tRNA sequencing data, it also, because that's a sequencing data, we can have a depth as well, the read numbers as well. That becomes promoter strengths as well. So we can have a promoter sequence and strength information. Using those, we were able to make CNN models to predict how strong each promoter would be in the cyanobacteria. So using these generated promoters, we rank them from 1 to 10,000 rank. And then we picked some of those candidates, and we did the evaluation using the evaluation method that we set up. And this is the yellow candidates are actually generated promoters. The gray things are just dummy sequences with the same length. This green is the promoters coming from the cyanobacteria, especially this promoter is a commonly used promoter. We can, as you can see here, we can easily generate promoters showing higher expression levels and also having a different scale of the promoter strength. When we apply this to in the cell, and although the length is slightly different, but we still, with the promoters are active in the cellular system, meaning that we can generate promoters for non-model organisms quite easily through this method. Okay, and also this is the second part regarding the molecular diagnostics using some of these circuit elements. And as you all experienced the COVID-19, you guys probably visited this kind of the screening facilities. The reason we visited the screening facility is that the screening facility, we use the standard method of the screening COVID-19 is based on the RT-PCR. So how does RT-PCR works is that once you go to the screening facility, the guys in the facility takes the swab of your samples. Then they put that into a red delivery liquid. Then they collect them and send them to a centralized institute to examine this. Then in a centralized facility, what they do is they take the RNA out and they do the reverse transcription and RT-PCR, and then they can figure out whether that's a positive or negative. So as you know, this is a complicated procedures, and also lots of the core and accessory instrument is involved. That's why we need some skilled people to run these operations, right? But if there's any method, we can simplify this. For example, if we focus on this single-strand RNA directly and somehow detect the RNA directly, then we can probably use these procedures. We can simplify the procedures. And also maybe if that method is robust to the impurities, we can probably use the red liquid that we used to deliver our swab samples directly, right? So what we did was we made different proof sets to detect this target RNA. So we made these two different proof, and each has a hybridization sequence complementary to target RNA, meaning that in the presence of the target RNA, these two proof binds to each other. By the way, we made these two proof locate right next to each other. And then what happens is that these two proof will be ligated based on this ligase, so that now we can have this one connected one proof. So now what happens is that we intentionally add here a T7 promoter site so that T7 RNA polymerase can access here so that it will generate a transcript. And in green, what we put is a aptamer template sequence so that once it is transcribed, then we can immediately have this kind of the fluorescence aptamer. In the presence of this dye, we can have fluorescences. So how that works is that if there's a nothing target exists, then no reaction happens. But in the presence of target, then the series of reaction happens so that we can have a fluorescences. One interesting thing is that because this RNA polymerase can transcribe these template sequences, which can generate identical sequence to the target RNA, meaning that we can amplify these reactions. Using this, so that was just the design, and to see if that works, we add all components for ligation and transcription and also the aptamer reactions all together and and also did some optimization of the buffers and everything. We were able to see some signals by having these target sequences. Then we diluted down to a 0.1 attomolar, see how low we can detect, and also how much time we need to discriminate these RNAs. And not only the SARS-CoV-2 virus, but also other viruses and also other bacteria even, we can target the mRNA or rRNA as well. We demonstrated also because the method to generate the proof is quite easy, so we try to target different bacteria and viruses, see how it works. And as you can see here, for the bacteria, different viruses, they had all similar kind of the sensitivity level, meaning that we can apply to different organisms. Then what we did was, as I said earlier, because we knew that by doing these procedures, we thought our method is quite robust to the different kind of impurities. For example, let's say it was quite robust to the cell lysate. So we thought maybe it's quite compatible with the other reagents. So what we did was we took the nasopharyngeal swab sample a little bit and applied the thermal lysis there. And also, along with that, we also applied the chemical lysis to directly break open the virus. Then we applied our reagent there, see whether we can get the fluorescences. And as you can see here, the positive sample is nicely discriminated from the negative sample, saying that the direct use of the swab samples is also applied as well. And because the method is quite simple, we can make a very simple devices that can read only fluorescences, and taking the nozzle printer swab sample directly, we were able to generate this kind of the flows, and we were able to make this kind of devices that can be very easily and cheaply developed. Also, another focus we had was that when we had a lot of variants, it was a problematic because it takes many days to sequence that out. So we thought because when we use the NGS to discriminate those kind of variants, then first of all, that costs a lot. Second, it takes a few days. So that we thought that's a problem, but we thought it can be solved by our method because our method is based on the ligation, and the ligation is a typical way of differentiating different bases. So what we did was we generate, for example, here this original sequence, and this is the alpha variant, and only the difference is this base C to U. So we generated two different sets of the proof, one detecting this original, the other detecting this alpha, and then see whether this each proof can discriminate the one from the others. For example, when we apply this proof for the original sequence to original RNA, then we can have this high signal. But when we apply this to alpha sequence, then we can have very low signals, which is almost background signals. And also we do this in parallel. We do this with a different sets of proof. Then we were able to see the other way around result. So meaning that we can use this to differentiate the SNPs. And then we found that works. And also what we thought was that this SNP discrimination is only required after we do the RT-PCR, and only positive can be applied to this kind of the discrimination. So we made a method to discriminate the SNPs in DNA sequences, amplified DNA sequences as well. So we use a directly RT-qPCR resultant or the RT-RPA resultant, see if whether from this double-strand DNA we can discriminate two different SNPs or not. So to aid that, to help to open two different strands together, what we did was we applied some DNA sequences complementary to each other, and that's what we call intruder DNA sequences. So with the help of the intruder sequences, we were able to make some rooms for our proof. That way, we were able to discriminate two different strands. For example, in this case, when we add intruder DNAs, shows much higher level of the fluorescences compared to the one we didn't have, the intruder sets. Then using this method, we tried to discriminate two different bases. This is 4 different cases, and it worked well. Within 20 minutes, we were able to discriminate the 2 different bases. And also, because the fluorescence fluorometer is cannot be quite fieldable technology, what we did was we made we hook up different visualization method. That's using LFA, lateral flow strip assay method. So to do that, what we did was we altered the reporter regions, and instead of having RNA aptamer template sequences, we generate this proof complementary sequence here, so that once the transcript generated, then this proof, which one side has a biotin, the other side has a FAM sequences. So in the presence of this, in the presence of this activity, then ligation and transcription goes, then we can have a biotin FAM can be aligned joined together so that we can have this gold nanoparticle can bind, gold nanoparticle can bind to the FEM side, and this which is connected to the biotin side. The biotin is captured by the immobilized streptavidin. So that's the logic. So when we apply this to discriminating different mutations, as you can see here, we were able to discriminate nicely for the different SNPs, and this is the actual results. And then what we did was we applied the clinical samples here too. So with the 4 negative samples, here we have 20 positive samples. 20 positive samples, we applied each different proof sets, whether to see whether we can discriminate these two. Unluckily, we didn't have any mutation for this regular base, but all 20 positive showed this N501Y sequence, indicating that, but it actually, when we sequenced that out, all 20 was these mutations. So meaning that we were able to discriminate this with even naked eyes using these lateral flow strips. So that's what we have. That's our progress regarding this SNP discrimination. With that, I'd like to thank all my group members. Highlighted students are actually participated some of this work, and these are collaborations. And also, I'd like to thank all funding sources. Thank you for your attention.
Thank you so much. It's such a nice talk. I would ask Martina whether she has question or comment first.
Thank you. Yeah, that was a very, very interesting talk. Thank you. It was very elegant work.
Thank you so much.
Just a question about kind of what you see yourself doing with it next. Like, where do you envision taking this next?
So you mean for the last part?
Mm-hmm. Yes.
So in terms of the mutation, I think we can apply this to discriminate the antibiotics. Or, I mean, this example is to discriminate the RNA viruses. And I think RNA viruses also good examples. And I think since this can be applied to not only RNA, DNA, any kinds of nucleotide SNPs, this can be developed into many different things. But for now, this is targeted to the RNA viruses.
Okay. Thank you. I appreciate.
Thank you.
So the Martina, do you have any other thing?
No, that was really my main question. I
Okay.
have, of course, it was interesting, but I'll
Sure.
let others go.
So I will ask the relevant question. So especially the last part that the first first part and second one is also interesting, but I know those work because I working on those things a lot. Last part I also working on. Recently I started organoid sensor using DNA, RNA as a target molecule to sense. But one bigger question to myself and to you, that is my, the only bigger question to everybody working on this field. So we sort of claim, pandemic is over. But who knows? I mean, we may have the second pandemic,
Right.
in near future. And then I see a lot of people now got COVID, after gathering, especially semester start, and then a lot of people get COVID. So to prevent or prepare for the next pandemic, and then cope with those kind of problem very quickly, because we now have experience, but the key thing we need to do is in timely manner rather than waiting several months to prepare those kind of things. So to get there, what is remaining challenges, and then how you gonna overcome that challenges in terms of speed response? That's my question.
In terms of the speed, what we need actually is the DNA sequence.
Uh-huh.
We need to know, we need to do the sequencing first.
Right. But
And
sequencing
once
we
we
can
have
do
this,
easily.
right. Once sequencing is done, we
Uh-huh.
can prepare the oligonucleotide very easily. So I think, just within a few days, we
Mhm.
can test out, see if, at least we can prepare different sets so that within a few days, we can generate a proper sets for the discrimination.
I
That's
see.
what I.
So that means, I mean, that is more likely diagnostic, and then you are saying diagnostic would be we can do quickly.
Right,
Yeah,
right,
but but technology development is one thing, but mass production of the diagnostic kit is another thing. But do
I
we,
see.
are we expecting any more challenges in terms of the distribution or, cold chain, distribution, etc.? Because that is another thing, because
That's
we
right.
didn't have
Yeah,
the kit until, I don't know, many months later, and COVID
right,
was, pandemic. But there is one question, but another question is how to make the vaccine very quickly. But you are not the vaccine expert, so I'm not asking that question to you. But in general, the speedy, response is very
right.
important. And
That's
then,
right. You're right.
how do you have any comment on mass production and distribution? How to do that quickly?
Uh, that's I think some part that beyond my knowledge, but
Sure.
based on my experience with all the you know the the, the government funded project,
Mm-hmm.
and we actually need to do a little bit of the we need to use some GMP facilities and
Mm-hmm.
all that. I think making, you're right, new kit is not quite as easy as we think, and we need to huddle the regulations as well. We need to get some permission to use, and we need to get some, for example, FDA approval
Mm-hmm.
before release this product, right?
Mm-hmm.
So that's, I think, one challenge we need to overcome. But before what we need to do is even for RT-PCR,
Mm-hmm.
the approval of the new proof is needed.
Uh-huh.
So for this kind of new technology, it's even harder. It
Mm-hmm.
takes many years, probably, if we go just a regular track. Maybe that's probably the point. Maybe later on, if this happens again, then it may be difficult to decide, but we need probably a faster track
Sure.
type of the approval regarding this kind of
Yeah,
uh,
that's
new method.
wonderful.
Yeah.
That's the good answer. So that might be the good way to go. I mean, you know, pandemic, everything was bad, except, because of pandemic, the technology advances is so, fast, because everyone just working on that one problem, and then we advance a lot of technology because of the pandemic. So we learn from the history, we learn from those kind of experience. So I hopefully next one, we, you know, address the problem very quickly than, several months or 1 year or even longer. So
Right.
now I see, let me check whether we have the question from audience. I don't see anything. So, and then also I see the 11:08 Central Time. That is over, a little bit over. So let's close.
Oh.
And then I believe the Jeong Wook need to go to bed. So the
Thank you so much for invitation and
sure.
preparing.
Yeah, I'll close. So thank you all for joining and staying today. We'll meet again on September 19, Thursday, the same time, the same Zoom link, we'll have the Professor Lydia Contreras at University of Texas at Austin and Dr. Akos Naiges at Harvard Medical School. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us, and I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Just give me 1 second. Stop recording.